Okay. So still people are joining. We have 173 on board. Can you guys see the presentation?
Yes, Vinay. Vinay, should we switch on the recording?
Yes. All right. Good evening, everyone joining from India and the folks joining from the US. Good evening to you. Good morning to you all. Thank you for joining. And it's so overwhelming and joyful to welcome you all for this new cohort of fall 2023 for cell biology and neurobiology course. And I just wanted to walk you guys through before Dr. Abhijit Majumder gives his introductory inaugural lecture for this course. I just wanted to walk you through with a little bit of background on how SciencePer was formed and what is the idea behind SciencePer. And yeah, so I'll talk about a little bit on the missions and the selection of the cohorts and who are the course directors and the highlights of the course. And also, we'll let the course director speak. And, we have the special seminar, inaugural seminar by Dr. Majumder, and we'll conclude after that. And coming to the idea of SciencePer, it is, as you all see, it is a non-profit teaching platform committed to provide quality education to all. We kind of put together this platform with a gap that we identified, that there are a lot of students importantly from the public education system in India and elsewhere who actually are very aspirational to pursue science careers, but they don't have that kind of quality education that they can access. It is only accessible to some of those people who are in the metros or in urban settings. But for the people who are not in the urban settings, it's been a big challenge for them. So we having identified that gap, friends who came along with us, enabled this platform to be a real one, to be an effective strategy to create a SciencePer platform that is centered on the problem solving and critical thinking by teaching from the research perspective in identifying a problem, devising solutions, and also how to implement methods to answer those questions. So this is the main idea of the platform. That's why we offer these courses for free. But these courses, we select the students with a very thorough process, which all of the students who are here have experienced that. So just to give you guys a sense of the applications that we have received this time, we have received applications from 10 different countries in addition to India. So you can see we have received applications from South American countries, some African countries, and also some Middle East countries. Of course, from our neighboring countries as well, from Pakistan, Bangladesh, and Sri Lanka as well. So this is a kind of a reflection how SciencePer is growing. Till now we had offered the courses only for Indian students, but now it has become an international platform. And as I told you, the application process is very stringent, and the selection process is very stringent, and we have got 400 applications for both the courses, and we have selected about 260 applicants for interviewing them. Thanks to all the panelists who are sitting here. Without you, this would have not possible. Would have been possible. Every candidate is interviewed for 15 minutes. That doesn't mean that we are kind of grilling the candidates and knowing the asking difficult questions. No, that's not the idea of SciencePer. SciencePer, being a non-profit initiative, not charging a single pie to the student, we wanted to see on whom we are investing this time. What is their motto on applying to these courses? Would they fit to our mission? And are they? Is there an opportunity for SciencePer to mentor them? So that is the main idea of conducting these interviews. In such a way, we have interviewed about 260 applicants. Out of them, we have selected close to 200 students, with over 90 students per cohort this time. And I'm really proud to say that we have over 55% women applicants in these cohorts. And this way, SciencePer also supports a little bit for the women STEM. Also, this time we have 20% of international candidate pool. And as I told you, the cell biology course has 98 students with 13 instructors teaching 25 classes, and supported by 18 teaching assistants, and adding 7 special seminars with from our friends who are working all across the globe. This is just to give you a sense of what is our instructor pool, like where do we get the instructors from? So we have instructors from all different universities in the West here, also from US, from Europe, and also I just wanted to also point that neurobiology course has a similar number of students, with close to 20 instructors teaching 35 sessions, with again 7 teaching assistants, with 4 special seminars conducted. And we have instructors coming all from different universities here in the New England area, also from France, also from different universities here in the US. With that, we'll go to Dr. Abhijit Majumdar's lecture. Before that, I would like to call upon Parijat, who is one of the course directors for cell biology. I'm sorry, I missed to mention this. So this year, Parijat, Anup, Rajan, and then Sanjeev are acting as course directors. Can you all say hi, Anup, Parijat, Rajan, and Sanjeev?
Hello.
Hello.
Hello, welcome everyone.
Hello.
Hi.
And Vinay, I think you should also mention the efforts of the TAs, which we are going to have this year. 8 TAs.
Absolutely.
And also Garima, because she has been instrumental in getting everything in place behind the scenes. So
there is time for that, and I'll call upon you guys. Why don't you take like a minute or so and introduce the course from your perspective before Parijat introduces Dr. Majumdar. I'll also call upon neurobiology instructors, course directors. Some of you, Abhijit, Anup, Rajan, and Sanjeev, and Parijat.
I think I have done this in the past, so maybe it's good time for either Rajan or Sanjeev to do this, considering they are new this time.
Yeah, so I can go first quickly. So hello everyone. I'm Sanjeev Sharma. I did my PhD from NCBS, which is National Center for Biological Sciences in Bangalore, and then moved on to do a postdoc here at Weill Cornell Medicine, New York. So, I mean, this year it's been a great pleasure acting as a director for the Cell Bio course. I have been part of the Cell Bio course ever since it started 3 years back. And I think all the instructors who have been part of it have have found it a really great experience in reaching out to younger students and interact with them, motivate them, mentor them in whichever way we can, and try and make um, things possible for them. So that's kind of what we've been aiming at through this course. And the idea has been always to try and reach out to newer and newer instructors who can take and introduce different kinds of topics every year. So the topics have always been slightly getting modified every year as the course runs. So that's another thing that happens with the course.
Yeah. Hi, everyone. I'm Rajan.
Yeah, so it's great to
Sanjeev
have
and
you all
I-
here, and
yes.
thanks.
Yeah, Sanjeev and I, we were from the same lab, we did PhD in the same place. Right now, I'm postdoc at Brown University Neuroscience. And just to add a few more points to what Sanjeev said, of course, biology is a very dynamic process, and we want to, convey that through this course that the field of cell biology is continuously evolving, and we would like to give you perspectives about different aspects of cell biology. If some of you are interested in pursuing something long term, we want to just give you a flavor of what it would look like from people who are actually doing the research. So that's one thing. And finally, the idea is to encourage all of you to understand that learning can be only complete once you interact and ask questions. So we also hope these courses will be highly interactive, where there should not be any inhibition in asking questions. So we want to try and have a class which is highly interactive, so that we can all question, think, and get to an answer together. So probably that's the idea. Let's see if we can achieve it or not, but yes.
Thank you, Rajan. I'll also call upon Aishwarya and Mostafizur, who are course directors for the neurobiology course this semester, to introduce yourself and give a little bit of introduction on the course.
Hello, everyone. I'm Aishwarya Krishnamurthy. I have finished recently my PhD in neuroscience from Brandeis University in Waltham. And I think it's super exciting to see that we have a huge cohort this time for both cell biology and neurobiology. And it's wonderful to be able to bring so many people from different countries together. And we do see that there is a lot of effort put in to get each one of you. And the idea behind, again, the neurobiology course also is to have a very, very interactive session and to be able to learn, for us to be able to teach and also learn from all of you. So we are hoping to have a very interactive and wonderful neurobiology course. Welcome all again to Science Spur.
Thank you. Thank you, Aishwarya. Thank you, Rajan. Thank you, Sanjeev. Now I'll let Parijat to come over and introduce herself, and then also introduce Dr. Majumder, the guest speaker of the day.
Wow, class to education. Online class.
Hello. Am I audible?
Yes.
Yeah, Parijat.
Okay. Hi everyone. Welcome to all the students, all the TAs, all the instructors. Yeah, everybody aboard, and a very big shout out to Vinay and Nag. Nag hasn't spoken a word today, but yes, a big shout out to them for having started Science Fair and having brought us all on board for teaching and all the, yeah, it has been a wonderful journey ever since it started. And I would start by welcoming our guest speaker, Abhijit Majumder. I will say a little bit about his career trajectory because it's really an interesting career path and a very interdisciplinary career path that is something to be appreciated in biological research today. And we need more and more of such kind of people in research to advance biological research. So Professor Majumder, he's a professor in IIT Bombay in Chemical Engineering Department. And after completing his bachelor's degree in NIT in Chemical Engineering, he did an MTech in Cryogenic Engineering and continued to work in soft materials in IIT Kanpur for his PhD. After which he transitioned to studying stem cells with Professor Jyotsna Dhawan, and he was during that stint in NCBS in STEM. And that's basically where I happened to meet Abhijit, and I have been following his career trajectory ever since. So when science first started and when we were discussing who to get for a guest lecture, it seemed like for many different reasons, he seemed to be an ideal person who could come and be a part of the course and basically deliver the opening lecture. He has a bit more about him. He has won several awards. I'm going to embarrass him by saying a couple of them. He got the DBT Wellcome Trust India Alliance Early Career Fellowship and even the India Young Investigators Award. He's a very inspiring and I gather a pretty popular teacher. And he leads a vibrant lab that works on various topics of mechanobiology. He not only studies fundamental questions, but also basically more on the application side of tissue engineering and delivering engineering cell delivery systems, which can advance fields like stem cell therapeutics and cancer therapeutics. So yeah, welcome Abhijit. It was really nice that you accepted our invitation and agreed to give a talk in this platform. I also admire him for his attempts at science outreach and that he actually makes an effort to not just conduct research, but also spread scientific awareness in general to people. So yes, it's wonderful to have you here and look forward to hearing. We have taken up quite a bit of time advertising ourselves. But yes, it is really an honor to have you on the platform. And yes, now the stage is all yours. Thank you.
Thank you, Parijat, for this nice introduction. And as Parijat just has mentioned that I did my postdoc from NCBS Bangalore, and there I met Parijat and Anup. So we had a kind of good time there. And as Parijat said that by training, I am a chemical engineer, but now my lab is trying to do a few things in biology. And all of this biology journey started when I was in NCBS. And actually people like Anup and like the PhD student and the postdocs there, they actually taught me the few basics of biology. So and thank you all for having me here. I mean, this is a fantastic effort. And I must say that when I got that first invitation, I thought the name is Science Pur, like the Dholakpur. So, it is a kind of a fantasy land. But yes, it is Spar, but obviously anything at Spar is also a fantasy land. So let us have a little bit of that journey, and I will share my journey as well as my research. So is my screen visible?
Yes.
Great.
Yes.
Thank you. So let me just try to adjust it a little bit. Okay. So as you can see that the name of this talk or the discussion that we will have here, so that will be on something called mechanobiology. So I understand that all of you are, people who are interested in biology, the students of biology. And I'm sure that as far as the biology is concerned, probably all of you know more than me here. But still, I would like to kind of discuss this particular aspect, which might be a new aspect for many of you. So, but before I start the discussion, let me talk a little bit about what my lab is doing. And the screens are not forwarding. Going ahead. Okay, fine. So as as you can see, my lab name is MLab, and M stands for Materials, Mechanics, and Microfluidics. So we try to work on in these three aspects. So these three are the building blocks of our effort that we are trying to do in our lab. Where is the highlighter? Yeah, so these are the building blocks: materials, mechanics, and the microfluidics. And on basis of these three building blocks, we are trying to understand some fundamental questions related to mechanics in stem cells and cancers and some other relevant biological system. And as Abhijit has said, not just doing the fundamental aspect. Based on that knowledge, we are also trying to develop some affordable technology along with my collaborators and some device that is known as organ on chip. And these are the few things that we are trying to do. In the recent past, we have kind of published a couple of papers, and these are something that we have tried to do to understand the fundamentals of mechanobiology, and then some of the things in terms of developing some technology. And these are some organ on chip, the book chapters, etc. So if any of you are interested, you are free to drop me an email, and I will be happy to share our papers or book chapters if you if you want to know more. So with this, other than this, again, some other examples, as I say, we are working on microfluidics. So some kind of microfluidic chip to create drug gradient, some kind of blood vessels on chip. Again, another, to create the drug gradient concentration, which can be used for testing the drug or some toxicology research, to study how some kind of surface structure, so this is a microscale structure, may influence cellular alignment, because in our body cells are not this way haphazardly placed, they are very nicely aligned. So that of some kind of study, and some devices to make some spheroid three-dimensional cell aggregate known as spheroids, organoids, or tumoroids. So these are the various different things that our lab is doing, just to give you an overall idea that what all you may do, this is just a fraction of what all you can do in this area of mechanobiology or the biomaterials. So with this small introduction, let me now start our discussion on these mechanobiology aspect. So when we talk about the mechanical or geometric cues, so this is essentially various different things that is together in our body, which may control the cellular behavior, okay, the cell functions. And these, obviously, the biochemical signals are important, but along with that, lot of the different mechanical signals, such as maybe substrate rigidity, fluid flow, some kind of surface property, if there is any kind of pattern on the, in your 3D structure, all of these mechanical or geometric cues are known to govern cell function and their behavior. So this is the kind of the what we understand from these mechanical or geometric cues, or what all different possible mechanical cues are there. So, but then why do we need to know, or why do we need to study this? Okay, so why do we need to study this? So I will start with a kind of an I would say assumption. It is a philosophical question, you can say. And I will start with that assumption, philosophical assumptions, that we, the multicellular organisms, are better than unicellular organisms. Okay, I mean, we can have debate whether we are better than bacteria or not, but let us, take that assumptions that we are better than them. So now, when we talk about these multicellular organisms, so say for example, us in our body. So how many cells we have in our body? So if it was in-person talk, generally here I stop and I ask the students to give the reply, but here I will kind of give the give the answer to you, that we have technical glitch, again not moving forward, but anyway. Let's try. Okay, yeah. So we have 37 trillion cells in our body. It is a kind of a calculated number, obviously, not a very fixed number. And how big is 37 trillion? This number is, just to give you a comparison. The Milky Way galaxy has 100 billion stars, or like 0.1 trillion stars. And so we have 370 times more cells in our body than the stars are there in the Milky Way galaxy. So clearly, when we have so many members in our body, and if we again take the assumptions that we are better than the unicellular organisms, all of these cells, they need to talk to each other. They need to work together. They need to work in synchrony, right? My brain needs to think, it needs to send the signal to my muscles, my muscle needs to move accordingly. So everything needs to follow a planned program. And for that, they need to communicate or signal with each other. And that's why when we look at the human body, it is not just a lump of the cells. It is always very nicely, intricately structured. Whether you take it, the heart, skin, eye, anything in your body, it is not just the cells. It is how the cells are arranged in their 3D microenvironment that kind of governs how they will function. So clearly, while the existence of individual cells are important, also it is important what kind of signals they get from the surroundings, how they are arranged, and in which environment they are in. So if we need to understand the cellular behavior, just studying cell in isolation will give us only a very limited knowledge. And to understand, the biology in its full glory, we also need to understand the microenvironment where they are in. So as I was talking about, that the signaling, the cell signaling, cell matrix signaling, the kind of signaling they get from their surroundings, all of these things are very important, right? So if I think of that, why signaling is important? Signaling is important, as you can see here, some example, that to communicate. Just giving one simple gesture or just some change of light, I can give signals to thousands of cars whether to move or stop. And so when we talk about that, this signaling, meaning is that it should have some basic features. And what are those features or the requirements? Requirements are that, how to get rid of this thing, I don't know. Anyway, the requirement is that Say, for example, if I take off these traffic signals, the person who is driving should be able to receive the signal, right? If the person is sleeping or looking at her phone, maybe he or she will not be able to take the signals, right? Then there should be set rules that what to do with the signals. So meaning is that when it is a red light, the set rule is that you need to stop. So meaning is that there should be the process to decode the signals. So first part is that sensing or receiving the signals, then decode the signals. And then as a driver, when I see the red light, I should be able to press the brake. So, and the brake should work. So I should have the capability to process the signal into action. So this part is called the processing and transduction. So all of these things are important when we talk about the mechanobiology, mechanosignals, mechanotransductions, etc. So when we look at the cellular system, as all of you know already, that we have all this set of various different chemical signals that is happening in our body. That is paracrine signal, autocrine signal, endocrine signal, all these various different signaling process that is going on in our body. And these chemical signals are based on mainly soluble and diffusible molecules. And again, as I said, the system should be able to receive the signal. There should be set rule when I get the signal, what should be done, and system should be able to give the response. So when I look at a chemical signals, it is just one particular pathway, and you can see how complex it can be. So these are the chemical signals that is happening in our body. Now we are getting into the actual part of today's talk, that is the mechanobiology or the mechanosignals. So as the name suggests, mechano. So mechano meaning mechanics related. And like what is mechanics? Mechanics is something that is related to force. And when we talk about force, obviously we need to talk about Newton. So essentially, in it, so the and like when we talk about the force, so what will happen with the force? We can push, we can pull, we can deform something, we can change the shape, and we can move something in motion, or we can stop something which was already in motion. So any signals which is based on any of these things, which is based on force, is called mechanosignals. And understanding how the cells receive those mechanosignals, how do they change that into a biochemical signals, and how do they respond to that signals, is the study of mechanobiology. So to give you one example, the most common example is hearing. I'm talking, you are able to hear my voice because in your ear, you have these cells which have these cilia, hair-like structures extended from the cell surface. When the sound wave goes, these cilia, they bend and they are attached with these spring-like proteins, which now when they bend, they get stretched, they apply force. Because of this force, these ion channels here gets opened, which allow the potassium signal to come in, and then the whole signaling process that happens in our body, or I mean, that happens in hearing, in the process of hearing. So essentially, when I am talking and you are listening to my talk, the essence is there is the mechanosignals. And there are plenty of such examples. I can tell you another example is obviously the touch. Touch is another very potent mechanosignals, which can send the signals of love, anger, hatred, and a lot of the different things, passion, and lot of many things. So the central question in my lab is, what is the role of mechanics in different cellular context. And that is what we are trying to understand. So again, as I said, that if this was that in-person talk, or if I was giving the talk in my classroom, I would stop here and I will say, take a deep breath. So when you take a deep breath, you are expanding your chest, your lung is expanding, and again, the whole mechanobiology is in function. So the question here one may ask, that when we talk about mechanosignals and mechanobiology, is it only for this some specialized cells, like our cells in our ear, or the sensation of touching, or the muscle cells in my rib cage, or is it more universal? So the one of the major understanding that we got, and so in 1980, this Harris et al., this paper came in Science, and it is a very, very interesting paper. Okay, a fantastic paper. What they have shown, and I personally call it that it is a first giant leap in our understanding of mechanobiology. It is my own personal kind of calling. It not necessarily everyone will agree to it, but anyway. So what they have done, they have taken a very polymer, which is like a thick oil. Then they expose that polymer to UV light just for a few seconds, so that just top layer gets cross-linked. Okay, you can imagine that on top of the milk, you get a very thin solid crust. Then they have cultured cells on them, that solid crust, which is essentially floating on a very highly viscous, very highly viscous oil-like polymer. And you can see here that the cells, they apply force on that rigid substrate, and it creates wrinkles. So from this wrinkle, they could conclude that yes, the cells are applying force on the substrate. You can just kind of quickly do that. You can apply force on your skin on your hand, or arm, or here, and you can see how applying force changes the wrinkles, creates and changes the wrinkle pattern. From this length of the wrinkles, they could do some back calculation, and they could get that how much force the cells are applying. So as why I say that this is the first giant leap, because this is the first experimental evidence that every adherent cell types, adherent meaning the cells which can attach to its surroundings, unlike say our RBCs. So all the adherent cells, they adhere to their surroundings, and then they apply force on their surroundings. This is the kind of learning from this paper. Later on, we got more such kind of results. I don't know where my computer is getting stuck in between, but fine. So we got more such papers. Here you can see it is from a 2010 papers. You can see here you have this small pillar-like structures. Okay, these pillars are of the micron scale. So one of my here is 40 micron, and diameter of these pillars might be 10 micron or 20 micron or so. So it is half of the size, diameter is half of mine here. And they are arranged like your toothbrush bristles. And when the cells, you put the cells on them, because cells adhere to these bristles and pull them, apply them, these bristles come inside. You can see that they get bent. Now, what you can conclude from here? You can conclude three different things from here. Number one, cells do apply force after adhering to the substrate or to their surroundings. Number two, because the pillars are bent inside, so cells are applying contractile inward force. And third, from the bending of the pillar, I can use the solid mechanics calculation to calculate how much force the cell is applying on each of these pillars and how that force is distributed all over the cellular surface that is adhered to that surface or the substrate. So these are the kind of techniques that we use to know how much force different cell types are applying. Okay, so you may ask the question, why do they do that and what is the biological significance? So why do they do that? One answer here is that as to understand whether something is soft or stiff, what we do is we need to apply force on it, right? How do you understand something is soft or stiff? You apply certain amount of force, then you measure, if I apply this amount of force, how much this material is deforming. If it deforms more, I will say it is a soft material. And if it does not deform, I will say it is a rigid material. So the cells apply force on their surroundings to measure the rigidity of their surroundings. What is the biological significance? So this is another very important paper in the field of mechanobiology. Here, so it is done by Engler's group from 2000. I mean, the first author was Engler, came from Danish Discher's group, came in 2006. And what they have done, they have used these cells called mesenchymal stem cells. So these are adult stem cell types. It is available in our fat tissues, bone marrow, dental pulp, in various other locations. They have used bone marrow derived mesenchymal stem cells. And it was known that if I give them certain type of chemical signals, the cocktail, differentiation cocktail in the media, I can differentiate them into bone-like osteoblast, fat-like adipocytes, chondrocyte, that is the cartilage type, neuronal, or the muscle. All these 5 different differentiation process is possible in case of the mesenchymal stem cells. That was already known. But what was known is that this differentiation can only be driven by giving them chemical signals. What this group did, they put the cells on the substrate of different stiffness. And why now, as we were discussing at the very beginning, that the purpose of this science per course is that you also need to, I mean, just not to get the information, you also need to think, when people do research, why do they think the question in that direction? And here the thought process was that tissues in our body, different tissues, have different rigidities. Calcified bones are the stiffers, and then the collagen, muscle, smooth muscle, the stromal cells, endothelial cells, etc. Different tissues have tissue-specific rigidity. So this millions years of evolution made the different tissues of the tissue-specific rigidity. Has it done just for fun, or there actually something there, right? So to kind of that govern their question, that raise their curiosity, and they ask the question, okay, if I keep these mesenchymal stem cells on the substrate mimicking brain, which is the softest, or something like middle stiffness like muscle, or much stiffer like bone, how these stem cells will differentiate. And what they have found is that if you keep them on a very soft brain-like substrate, these mesenchymal stem cells start becoming neuron-like if they put on the muscle-like substrate with muscle-like stiffness or rigidity. They start to differentiate into muscle-like differentiation, muscle-like features. And if you put them on a very rigid substrate, they start to become bone-like or osteoblast-like. So essentially what they have shown that the stem cells, which have many different ways to go in front of them, you can control their fate simply by controlling mechanical environment of their surroundings or just by controlling mechanobiology. So how does it work? So this example is typically from the muscle, but not necessarily just the muscle. In your body, or sorry, in each cells, you have actins, so actin stress fibers, and then the myosins. So these myosins get adhered to these actins, and then they pull the actin. And now these chemical cues, as I said, say for example, the stem cell differentiation cues and the mechanical cues that the cells get from their surroundings, they together give the cell microenvironment. So what is mechanobiology? Force interaction between cell and its surroundings. So if I think of this is my cell membrane, and this is the cell surroundings or the matrix, and this side is the cell. This is the focal adhesion, the adhesion points with which the cells adhere to its surroundings. And one end of the adhesion points are attached with the actin cytoskeleton, where you have the myosin motors. Myosin motors apply force on this actin cytoskeleton. This actin cytoskeleton apply force on the focal adhesion. Other side of the focal adhesions are attached with this ECM matrix. As a result, cell applied force results into deformation of this matrix. Depending on the matrix rigidity, it will depend less or more. Cell will sense that and it will behave accordingly. So this is essentially the core of this mechanical sensing or the mechanotransduction process. So as I have just said, cells apply force on matrix, then the matrix give the reaction force, Newton's third law. So this is cells applying force on, matrix applying force on cells. Also, say in case of our blood vessel, there can be blood flow, which can directly apply force on the cells like the fluid shear. So it is not a reaction force, it is an active force applied by the environment on the cells. And then cells sense these force, and they do some changes which are fast, take minutes to hours, such as assembly of the focal adhesion, maturation, that is these proteins which are here, using which cells attaches to their surroundings. They kind of mature, they grow, lots of more, they change their, their shape, and then lots of other proteins get involved, so they get matured. Then this actin cytoskeleton, this focal adhesion is attached with this rope-like structures, which essentially the skeleton of the bone, that becomes mature. That gives the cell its shape and the morphology, and that may also impinge on the nucleus, the control center of the cell, and may change its morphology as well. When all of these changes happens, that will also have a feedback, and the cells will change its force on the matrix. So there are some feedback loop involved. So this is the first response, and there will be a mechanotransduction process, which is a delayed response, and that we will see that the effect on cell cycle, how cells proliferate, cell survival, apoptosis, differentiation. I just gave you the example, cell migration. Essentially, you name any cellular activity, and there are research work which has shown that yes, these mechanical signals are important there. And when all of these things will happen, again, there will be a feedback loop. For example, when the stem cells will differentiate, they may change their shape. And as a result, they may change that the force that they were applying on their mechanical microenvironment. So this is what we try to understand in this understanding of the mechanobiology, that in short, how this microenvironment is influencing these various different cellular processes. How much force, which parameters that cell sends, what changes focal adhesion, etc., etc. So all of these various questions are various different positions that we ask. Okay, and why do we want to do this? Maybe we can design a biomaterial which may change, but I can just switch and I can kind of, direct a stem cell to differentiate into whichever way I want it to go. So again, just to continue with that question, why do we want to know this, right? So as you can see in this slide, that the life is complex, and in our body, the cell here, it is getting so many different types of different signals from its surroundings. Physical signals, soluble factors from their neighboring cells, extracellular matrix, it is all getting. When we do our cell culture in the lab on these Petri plates, most of these signals are missing there, right? And so many of the times, or I would say probably most of the times, a lot of our in vitro result, that is the result that we get during the, by culturing the cells on these Petri plates, they don't match when we do that in animal. And the result that we get from the animal, they don't match when we do the clinical trial, say for a drug or when we test in the human. And so that's why if today I start with 100 different, the molecules which may have ability or to cure any disease, which show some promise in curing some disease, after the clinical trial, starting from the in vitro to in vivo to human, just one drug that actually succeeds. So our success rate is very low because we don't understand the complexity of the system as a whole. Okay, and so for that, some people, including our lab, we are trying to make something like gut organ on chip, but that is not the, that is not what we will discuss today. So I will just again go back to this slide that I showed you earlier, that there are these various different mechanical and the geometric cues that is present in our body that the cells sense and they behave accordingly. And this is the study of the mechanobiology that our lab and many other labs are working on. So with this, I will take an intermission here. Interval, I have taken half an hour. And after this, I will give one specific example from our own research. So should I take some questions now or should I keep it for the end?
Yeah, sure. Go ahead and take questions. Yeah.
Okay. So if there is any question, please feel free to ask. This is popcorn time.
Hello. Hi,
Yeah, hi.
Abhijit. This is Sakshi.
Yeah, hi, Sakshi.
Yeah. So while you were explaining the slide where different differentiation of mesenchymal cells shows differentiation with respect to the substrate you're choosing. So if you can explain what kind of substrate you're talking about.
Okay. So here in this particular experiment, what they have used is polyacrylamide gel. So it's a polymer and you can change its rigidity, its mechanical property, soft versus stiff, by changing the cross-linking density. So they have used polyacrylamide gel, a 2D substrate. Then they have coated that substrate because cells will not adhere to that substrate. So they have coated that substrate with some kind of extracellular matrix protein, say collagen, fibronectin, laminin, and then they put the cells on them. Now the cells adhere to it, apply force, and depending on the cross-linking density, which governs the rigidity of the polyacrylamide gel, and according to the rigidity, the stem cells would differentiate.
Okay, all right. So my question was, so while differentiation won't only this substrate stiffness will only provide stress in such a way that it will help this differentiation to change shape. How it is helping them to change entirely to a different new type of differentiation without any chemical signaling?
Yes, so that's actually a very good question, and that is essentially the crux of the mechanotransduction. That it has been done a lot after that. That when these you give them to the different mechanical signals, they apply force, as I said, and then a lot of the different things happen in the cell, right? The first thing that will happen is that this focal adhesion, which are the first focal adhesion molecules are the integrins, that integrins will come together, they will cluster, then they will recruit other focal adhesion proteins, and then it will start a downstream signaling process. Okay, and then this downstream signaling process will invoke, so it will change the conformation of different proteins because of these force interactions, and that will invoke the different pathways, which will be, which will then cause the change in the epigenetics of the cells. Okay, say for example, I can just give you one example. For example, one transcription factor known as YAPTAs. So when cells are put in the stiffer substrate, these YAPTAs can go from cytoplasm into the nucleus. Because the nucleus also senses the force, nuclear pores gets opened, YAPTAs goes inside. Now it's a transcription factor. It will now invoke RUNX2, which is a gene required for osteogenic differentiation. So that's how, when you put the stem cells on a stiff substrate, it will go into osteogenic differentiation. That is just one mechanism. There are many other mechanisms how these, the mechanical signals get converted into biochemical signals.
Would you mind sharing some paper on related to this?
Definitely. So some of the references I have given while I am kind of talking, giving the talk. I can share, some of these papers with the organizers, and they can share with you. Definitely.
Cool. Thank you.
Thank you, Zepsi.
Great. I just wanted to say one thing to the students. Thank you for writing your questions in the chat, but we encourage you to speak out and also have your video on so that the speaker can see you, right?
Yeah.
Great opportunity
Yes.
to do that.
Students and instructors alike, if you can.
Excuse me. Good evening, sir. May I ask the question?
Hi, Idris.
Sir, my question is related to the same topic: substrate stiffness, director's stem cell differentiation. So my question was, how do you take concern of substrate used and mechanocycling used in system technology?
Idris, I actually could not follow. There was some noise. Can you please again repeat?
Sir, I have typed the question as well. My question was like, how we take the concern of substrate used and mechanocycling in system technology when we are going to form any organ?
When we are going to form any organ, how these mechanosignalings play a role? Is that
Yeah, exactly. And how we take the concern of substrate.
Okay, so I think it is a very open question. Depending on what we are trying to design or what we are trying to achieve, we often try to mimic the mechanical signals that is relevant to that particular physiological situation. Just to give you one example, suppose, say when people want to develop the blood vessels in vitro with the endothelial cells, it has been shown that if you have the fluid flow and the fluid shear stress, that actually help the endothelial cells to align and kind of to get the maturation to form the lumen structures. Okay, there are other examples. For example, in case of the cardiomyocyte differentiation, it has been shown that if you give them a cyclic strain, mimicking what they are supposed to experience in body, this cardiomyocyte differentiation process gets enhanced.
Okay, sir. Thank you.
Thank you. So let me move with the, there might be other questions, but let me move with the, the next part of my talk with one example from our group, our research, and I'll be happy to take more questions after that. Is that okay? Yeah.
Yeah, reserve all the questions for the end. We will have a long question and answer session if time permits.
Sure.
Yeah.
Intermission is over, popcorn time is over. Yes. So this is my journey, just a quick glimpse to my journey. That, and why I keep this slide also, because you are here, the students, you may, many people come to me with, a lot of the career guidance kind of question. And I tell them that, the boundaries are really fading. Okay. So, you can try, you can work, or in your undergrad level, you can study anything, and then you can shift the field if you have interest. So I learned soft mechanics in my PhD and the microfluidics, microfabrication. I had no experience of biology. So I did my PhD from Kanpur. Then I moved to Kanpur to Bangalore, because after the PhD, my question was that where I can possibly apply this knowledge of soft mechanics, microfluidics, microfabrication in biology. Because biology was kind of something that I always wanted to do. I wanted to become a medical professional, a doctor. I could not crack the medical joint, and I tried twice, and both the times I could clear the engineering joint. So the second time people said, okay, you just go for engineering. I said, okay, fine. But then I will finally, come back to biology this way or that way. And I will also put doctor in front of my name. So that happened. But, this was a long, tortuous journey. So when I joined Professor Jyotsna Dhawan's lab at Bangalore, and here I must thank Jyotsna, because not many people will take a postdoc in their lab without any knowledge of what the lab is doing. But Jyotsna was ready to, take chance. And so, I must thank her. So as I say, my journey was from soft mechanics to cell mechanics. So I did my PhD with Professor Animangshu Ghatak and Professor Ashutosh Sharma, and there I worked on understanding some adhesion in the context of some kind of in the context of microfabrication and adhesion in the context of biomimicry. And then I joined Jyotsna's lab at NCBS. And this was this lab and these people who literally taught me, how to do biology, literally holding my hand. And I am not kind of exaggerating, because if I just show you what, so I joined here in 2010 February, and what I wrote in my NCBS diary, that they used to give us a small notepad, and there is this thing from this notepad. And here on 8 Feb 2010, I am writing unknown terms, mesenchymal stem cells. And then after 13 years, I am explaining the stem cells to 190 people, right? So then cell cycle, myoblast, C2C12 myogenic cell, motility, the terms and topics I need to know. So when I joined Jyotsna's lab, my understanding was even lower than an UG intern. So all these people helped me to kind of, go through that process. So, and one of the major problem of in terms of changing the field was the jargons. As I said, the lost in translation. Jyotsna used to, laugh that for her. So she said that when we talk about in the biology, when we talk about, why my computer is getting hang, that she said stress and strain, are they two different things? I always thought stress and strain are the same. And I told Jyotsna that, and Jyotsna said, when we talk about stress, we mean this. And I told you, so you know, as an engineer, when I talk about stress, I mean this. And when they talk about X and Y, they meant this. And when I talked about X and Y, I meant this. So there was a wide, you know, the difference in terms of the way we talk. But then slowly, we learned each other's language and kind of went through. And from that, now our lab is working on various different aspects. And again, I am still learning. I'm a still learner in the field of biology, and I am learning from my collaborators as well as from my students who are kind of coming from various different backgrounds. So now I quickly give you one particular example that our lab has worked on in collaboration with Dr. Shilpi Dutt from Actric. And the work that I'm going to show is done by my student Pallavi, who already graduated, Ketki, who is still doing her PhD, going to graduate soon, and Anagha submitted her thesis from Shilpi's lab. So when we talk about the glioblastoma, it is a type of a brain cancer. And if you look these two graphs, this is very important, is that you see even in these last 50 years, the life expectancy has not changed much. It is still, if today someone is detected with glioblastoma or GBM, their life expectancy is hardly 1.5 to 2 years. And it is the, so per year we get 2.4 lakh brain tumors. Out of that, GBM is the most common form of the primary brain tumors. And as you can see, 5-year survival rate is like 3% or less. Median survival rate is just an year. Okay, so we have not really progressed in this field. For particular for this cancer, I mean, obviously we have made a lot of progress in this field, my mistake, but in terms of saving people's life, we have not progressed much in the last 50 years. We have done wonder in many other cancers, but this one is still a tough nut to crack. So when we look at these, this prognosis of the GBM, what happens is that first, someone is detected with GBM, they go through the chemotherapy, radiotherapy, maybe the surgical removal of the tumor. So this we call as the parent tumor or the primary tumor. Almost certainly in next 5 to 10 months, the tumor will come back because there will be some residual glioma cells and they will form a relapsed tumor. And when the relapsed tumor is formed, it is known that the relapsed tumor is much more aggressive than the parent tumor in terms of they are highly proliferative, they are highly invasive, they are drug resistant. So essentially, I can tell that if people are going under treatment, they are not dying of the parent tumor, but patients are dying of the relapsed tumor. So we really need to understand the biology of the relapsed tumor. Now for that, in Shilpi's lab, they have this model where they take some, say, cell line, the glioblastoma cell line, as well as the patient cells. They expose them to a certain, radiation, mimicking that the radiation that happens in the treatment. In your plate, all the cells die. If you look under the microscope, you will not see any cells, but somehow some cells survive. And in 10-14 days, the plate will be full with the new cells, which we call as the recurrent, the radio-recurrent or the relapsed cells. However, if I look at this parent and the relapsed cells, you can see they look very similar, not much different. When we look at the other cancer aggressiveness parameters, such as, say, proliferation, migration, invasion, so what we see here is that the parent and the recurrent cells have a little bit more migration, but not statistically significant. They have a little bit of more invasion, but not statistically significant. Why this migration and invasion is important? Because that will cause the tumor cells go from one part of the brain to the all other parts, and that is called the progression of the cancer, right? However, when we take this parent and the recurrent cells in vitro model and we put them in the mouse brain, we can clearly see that the in vitro cells have, recurrent cells have proliferated more, spread out more compared to the parent cells. There is a clear-cut difference. These green ones are my recurrent cells inside the mouse brain. So clearly, these recurrent cells and the parent cells behave differently when we put them in an in vivo mouse brain, but when we look at in vitro, they are behaving same. So if we need to study them in vitro, I need to create a model that can mimic the brain. So for that, in Shilpi's lab, they have, again, why we think in certain direction that I am trying to tell you, that they have taken U87 to different cell lines as well as patient sample. They have compared between parent cells and the radio recurrent cells and the relapse cells. And when they look at what is the major difference in their pathways, the three pathways that comes at the top, first one is the pathways in cancer, which is kind of, very obvious. But other two are the focal adhesion and ECM receptor interactions. That tells us that the major difference between the recurrent cells and the parent cells are lying how do they interact with their extracellular matrix and the surroundings. So we thought that when we are culturing the cells on these plastic Petri plates, they are much rigid. But brain is one of the softest tissue. So should we put the cells in a soft gel and to look at that whether we can recapitulate this aggressive nature of the recurrent cells? So I must tell here that we are not the first one to look into how the substrate stiffness influence GBM cells. There are other papers. However, we were the first one to compare the response of parent versus relapse cells to the substrate rigidity. So there we bring our novelty, our research question, our curiosity-driven question. So we are asking this question. Do the parent and radio-recurrent cells differ in their mechanosignaling mechanoresponse? And our second question is that, can we recapitulate the aggressive nature of the recurrent tumor in vitro? So what we have done, first, what experiment was already going on in Shilpi's lab, that is parent cells and the relapse cells, putting them on the plastic Petri plates, which are much stiffer, gigapascal, 10 to the power 6 Pascal range. Whereas brain is only 500 Pascal. So what we did, we put them on gel, which is 0.5 kilopascal. We measured the brain stiffness. We found that, okay, it is 500 Pascal in that range. So we make the gels, we put these parent and the relapse cells on these soft gels. So what we are comparing here? We are comparing here parent cells on plastic, recurrent cells on plastic, parent cells on brain-like stiffness, and the recurrent cells on brain-like stiffness. These four things we are comparing. So what we first observed, I'll quickly kind of go through here, that when I put them on plastic, if I look at here, the cells look very similar, parent and the recurrent. However, when we put them on the brain-like stiffness, in the parent population, I get all round cells, but in the recurrent population, I get 50% round cells and 50% elongated cells. So clearly, how they sense and respond to their substrate stiffness are different. Then we look at, so it is the cell spread area, etc. I will just skip that part. Then we looked at how much force these cells are applying to their surroundings. Again, we can see that these parent cells and the relapse cells, they apply different amount of force on their surrounding. And these circular and elongated cells, they also differ the amount of force they apply. Okay, so clearly this parent and the retrorecurrent cells, they differ in their mechanosignaling and the mechanoresponse. Next, our question is that, can we recapitulate the aggressive nature? So again, I will try to kind of remember what I mean by aggressive nature is proliferation, because if they proliferate more, they will increase in number. Then migration and invasion, the tumor will not be just one end, they will go everywhere. Drug resistance, and if I put the cells in the mouse brain, how big tumors they are forming. So these are our measure of the aggressive nature. So first we looked at the migration, as I said. Now you check here, this black is the parent cells, red are the relapse cells. When I culture them on plastic, their migrational velocity are absolutely the same. When I put them on 0.5 kPa brain-like stiffness, the relapse cells have much higher velocity, almost double the velocity than the parent cells. Then we look at the invasion. So to invasion, what the experiment is that, just if you don't know, just that we have a membrane here. Then here I have one media where I have some kind of growth factor to attract the cells. This side we have the cells without the growth factor. So the cells will, and there are some kind of ECM matrix. So cell which are initially here, they will invade and go to the other side of the matrix. So what we are doing here, we are putting the parent cells, relapse cells from plastic, from gel here, and observing how many cells are going from this side to the other side of the membrane. So when we look at the parent, the 0.5, you can see, even if you culture the parent cells on the brain-like stiffness, there are more number of cells that have gone the other side of the membrane. So clearly, culturing them on the brain-like stiffness makes them more invasive, give them more invasive nature. However, the dramatic change was that when we compared the relapse population, and what we have seen is that when we put the relapse population, you can see parent and relapsed culture on plastic, not much difference, relapse a little bit more. But when I put that on the, the culture, the relapse cells on the brain-like stiffness, you can see this side is full with cells. So essentially, it gives them an immense invading ability, just simply culturing them on a brain-like rigidity. So recurrent cells cultured on gel is more migratory and more invasive. So I'll skip this slide in interest of time. We also checked for the proliferation. Again, the same story, culturing them on plastic, parent and relapse, similar proliferative. But when you culture them on brain-like stiffness, relapse cells are much more proliferative than the parent cells. Then we looked at the drug resistance. We looked for a common drug used to treat GBM, temozolomide. And again, I will just repeat the same thing. If you look at here, I used 50 micromolar drug. If I compare the cells coming from plastic, parent and relapse, they are responding similarly when you are treating them with temozolomide. However, if I culture the cells on gel, then clearly the relapse cells cultured on gel, they are much more resistant. Meaning is that I'm culturing them for 72 hours on 50 micromolar drug, but 80% of the cells are surviving. Meaning is that your take-home message here is that if I do say, you just imagine temozolomide is a new drug and you are testing that in the lab, culturing the parent and the relapse cells on plastic, you will conclude that they are equally responsive. However, in reality, They are not. Relapse cells are known to be drug resistant, and that can be captured only when you grow them on brain-like stiffness. And then we put the cells in the mouse brain, and again the same story. The cells, the recurrent cells coming from the gel showed the most, most tumorogenic nature. So what we got here is that relapsed tumor cells grown on brain-like cell showed more aggressive phenotype. We have checked proliferation, migration, invasion, drug resistant, and high tumorigenicity. So essentially what we are concluding here is that to capture the clinical observation, that is that the relapsed GBM tumors are much more aggressive in nature, if you give them the pathophysiologically relevant mechanical environment, you can capture some of the aspect of the reality which you may miss if you culture them only on the plastic Petri plates, which is very different from their in vivo pathophysiological environment. So essentially what we have talked here today is that how mechanics and the cells, or which is kind of a physics and the biology, they interact with each other. As I told in between, when I joined Jyotsna's lab, as Jyotsna said, for them the stress was this, for me stress was this, but we have seen these two worlds are not really separate. They always talk to each other, and whichever may be your interest, but you can still cross the boundary, cross the border. And I would also like to make some geopolitical statement here, is that all these political and these boundaries and borders are meaningless. We need to look for a world where things go hand in hand. We will try to touch each other and make bonds. So with this, I would like to end my talk here. And this is all the people, my lab members. Earlier, when I started the lab, and this is how the lab is. I have lost some here, and some people have moved on their life. Some new people have joined. And the last part of the talk was from these people. These are all the grant agency. And as I was talking about, that the world will be much better place if we hold each other's hand. And so I will, use this proverb. I have read it internet. It is an African proverb. I don't know if it is correct or not, but the proverb is really good. If you want to go fast, go alone. But if you want to go far, go together. So I will again thank SciencePar for giving me this opportunity, and also for bringing people together with the aim to go far. Thank you. Thank you very much.
Yeah, thank you so much, Abhijit. That was a very nice talk and a wonderful way to conclude the talk. Yeah, and the floor is open for questions. And I do have a question. I'll go first. It is something very general. I really liked that how you gave sort of a sense of dimension in terms of hair size to the size of the pillars and then you gave a sense of numbers of cells by comparing to the numbers of stars in the galaxy. Can you give a sense of the force that cells can exert or generate in terms of some tangible examples, just for students and all of us to kind of relate?
Okay, so cells apply force in the range of piconewton to nanonewton. Okay, so now micro is 10 to the power minus 3, minus milli is 10 to the power minus 3, micro is 10 to the power minus 6, and pico is 10 to the power minus 9. And 1 Newton is the force that we need to lift up 1 kg of the weight. So you can think of that 10 to the power minus 9 of that kind of force that we are talking about.
Minus 9 would be nano, right? Pico would be
sorry?
12.
Sorry,
Minus
sorry,
9
nano.
would be
Sorry,
nano,
sorry.
okay.
That will be minus 9
Yeah.
is nano, and pico will be 10 to the power minus 12. You are right. Sorry. Yeah. And just to give you another kind of example is that if you take a eraser, the stiffness is in the megapascal, that is 10 to the power 6. Okay. Meaning is that to make a unit amount of deformation, I need to apply 10 to the power 6, unit strain, I need to apply 10 to the power 6 force in unit area, right? And so that is 10 to the power 6 Pascal. And I was talking about the brain, which is 500 Pascal. So 10 to the power 6 and the 500. So this is the kind of range we are talking about here.
Okay, thank you.
Okay, so Dr. Abhijit Majumder, I have a question about the organ chips that you said.
Sorry,
Organ-like chips.
some echo is happening here. Can you please again? Yeah, please.
About the organ chips that you said that will replicate the organs of the humans.
I don't know. It is breaking at my end, or others can hear. It is his voice is breaking at my end.
Yeah, even we can't clearly hear. If there is a problem, maybe you can write on the chat.
You may write in the chat box, and Parijat, you may read that question for me, please. And I see Samson has raised his hand, so I can take that question in between.
Yeah, thank you so much. Can you hear me?
Yeah, I can hear you. Thank you.
Okay, you have been able to highlight the tremendous effects of environmental rigidity on cellular functions. And then my question is there any degree of specificity with regards to this cellular differentiation if this differentiation is solely instigated by the rigidity of the cellular environment?
A degree of what?
Is there a degree of specificity in this cellular differentiation?
Specificity, yes.
If this differentiation is solely instigated by the rigidity of the cellular environment without chemical signaling.
Correct. Yes, absolutely. You are absolutely correct. That I have not gone that kind of detailed here. Is that when we talk about the stem cell differentiation, that's a very good question, Samson. That when we talk for the stem cell differentiation, it is not that 100% of the cells do differentiate. And also, differentiation is not just a black and white, a 0 and 1 thing, a binary thing. It goes to the multiple different intermediate steps, right? So that particular word that I talked about, they were talking about the lineage specification. That is the expression of some early genes. There we were not talking about the complete full range of differentiation process. And I may add here is that it has been shown by them as well as many other researchers later, is that the chemical signals and the mechanical signals, they actually can work in synchronization. And if you give both the signals in a particular direction, you can increase both in terms of what percentage of the cells are differentiating, as well as how far you can go in your differentiation process. So with mechanical signals alone, full range of the complete differentiation might not be possible. Thank you, Samson. It was a very nice question.
Thank you.
Parijat, can you please read the question if he has already typed?
Yes. Can you hear me? Yeah. So the question is, how could you tell a little bit about how can this matrix stiffness or physical factors be used in cancer treatment? Does a particular amount of force induce particular amount of cell membrane specific proteins? So I think these are probably two different questions. Yeah.
So I think the name was Santosh, or who was the question?
Yes. This was from Idris.
Yeah, Idris. Sorry. So Idris, if I start now talking on cancer, that will be another full talk. But I mean, I have given you one example of cancer there. Mechanics play a very, very important role in cancer. To understand that if I talk of a solid tumor, the first thing is that the sensation that it is stiffer than the surrounding matrix. So clearly, they secrete more ECM, the cancer cells, they secrete more ECM, particularly collagen, and then they make their environment collagen rich. They also secrete more MMPs, because you need to now think of that these tumor cells, to do the metastasis, they need to invade through these ECM structure. So they also secrete MMPs to cut and make path, but at the same time, they also secrete collagen to create path. So it is kind of, you are chopping the path as well as you are laying down the path so that you can move on. Right. Many of the cancer-related pathways have been shown. I mean, and in all type of solid cancers, that they have a strong correlation with the stiffness. Both in vivo and in vitro things have been shown. Whether it is breast cancer, brain cancer, cervical cancer, the cancerous tissues are almost all the time much more rigid than the normal tissue. And I mean, many of the stiffness related signaling process related to gene expression, protein expression, microRNA, long chain non-coding RNA, I mean, there are like plenty of work in this field. So the short answer to your question is that yes, mechanical signals impinge on cancer, and as it is said, cancer is a disease of seed and the soil, cell, as well as the microenvironment. So yes, microenvironment plays a very important role, and people are studying that in details.
So there is another question. There are a couple more questions that I will just go through quickly from the chat. Faizan has asked about the organ-like chips. I guess she means
organ on chip.
Organ on chip. And how will these chips work?
Okay. So again, I mean, that will be, again, as I said, another day of discussion. But so the Faizan, the name you said Faizan, right?
Faizan, yeah.
So Faizan, organ on chip that we are working on, they are not organs, okay? They are generally used particularly for the drug study or the toxicology study. So I will just give you one example, and probably that will help you to understand that what we are talking about here. One of the first organ on chip was the lung on chip, done in the Harvard. So what they did was that they took a membrane, a polymeric membrane with micron size holes. Now, if you think of the lungs, one side you have the blood vessels, that is endothelial cells, and another side you have the lungs, the epithelial cells. So in these polymeric membrane with small holes, one side they have endothelial cells, another side they have epithelial cells. Endothelial cells had media flow like blood, and the epithelial cell side had only air flow because that was mimicking the lung side. And then what was done is that in the endothelial side flow, you have the immune cells, and in the air side flow, you put some bugs which kind of known to infect the lungs. So when those bugs get attached to the epithelial cells, here in the endothelial side, the immune cells get activated, they latched on that location, they invade through those small holes and they kill those bugs. And then that particular group had also shown that if our lung is always expanding and kind of shrinking, right? There are the mechanical force. Now that polymeric membrane, if you now expand it and shrink it like your lung, the activities changes. So this is just one example of lung on chip. In our lab, for example, we are working on the placenta on chip. So there, one side we have the endothelial cells, again, another side we have epithelial cells, and we are trying to mimic the flow rate and the shear stress, etc., that is present in placenta. And we are trying to look at how, say, when the baby is growing in mother's womb, you want some of the things to pass the placental membrane, other things don't. So when you design a drug, if the drug is for the fetus or the placenta, you want the drug to cross the placental barrier. On the other hand, if the drug is for the mother, you don't want the drug to cross the placental barrier. So we are working on this placenta on chip, which we are now kind of trying to look at, that can this chip predict which drug will actually cause the placental barrier and not. So these are the kind of two examples that I could give you.
I think let's go through the questions of the hands that are raised. They can probably speak their questions out to you, and then we will go back to the chat questions. There are quite a few in the chat. So let's go through the questions from the students who want to ask verbally first.
Hello. I'm. Okay. Oh, go ahead. Oh, okay. Yeah. Thank you. I'm Navneet, and I have a very simple question.
Do you want to turn on your video we would like to see you when you're asking the question?
Oh, I'm in a very dark background actually, so you won't be able to see my face.
Okay,
that's fine.
no worries.
That's fine. Yeah, please go ahead.
Yeah.
Okay.
So yeah, my question is very simple, and it was a wonderful talk that you gave actually. So when we talk about stem cell differentiation, you basically told that there are two signals that the cell actually receives, a mechanical signal and a chemical signal. And you have been very clear about mechanical signals, about the megapascals of the substrate and all. But what kind of chemical signals are we talking about? And is there any difference between the chemical signals that we give to a stem cell in vitro, and is it any different chemical signals that a stem cell receives when it is in vivo? Is there any difference between those chemical signals? These are the questions that I have.
Okay, first I will tell you so when we say the chemical signals, we mean different types of, say, for example, growth factors or like other chemicals such as retinoic acid. So the cocktail is made with and these are very much specific for particular lineage differentiation, right? If you want, say like for example, insulin. So if you want a particular differentiation to happen, and that has been done, in years of research, that if you make this cocktail of, say, 4 different things, your cells will differentiate into, say, osteoblast or the adipocytes, etc. So when I say the chemical factors, I mean this kind of growth factors, hormones, some other chemicals, as I said, the retinoic acid, etc. Now, your next part of your question is that is it different in vivo versus in vitro? Definitely yes. In vivo, you get a milieu of thousands of different chemical signals in various different ways in a spatio-temporal manner. In vitro has been done, as I said, in the years of research, that what are the minimal requirement that we need to give to get a certain reasonable amount of differentiation. As I was also telling while answering, I think, Samson's question, that there is no, the in vitro differentiations are not complete. So, yeah.
Okay, a small question a follow-up question, a very small follow-up question. So how will this difference in the chemical cocktails that we give to the cell will result in the efficiency of the cell to take up signals on a long run? Because when we're doing it in vitro, we're giving a very limited amount of stress, and when it is in vivo, it's receiving millions of stress. So how will that affect the cell on a long term in receiving signals and giving a response to it? Is there any difference, or will the cell adapt?
I'm not sure if I have understood your question correctly, but so essentially you are asking that what is the difference between in vivo to in vitro in terms of these chemical signals, or?
No, you told that in vitro, when we are culturing cells, we give them a cocktail. And the signal that the cell receives from this cocktails is the minimal requirement compared to what it receives in vivo, where there will be like millions of stresses that the cell will be getting.
Very different,
so how does this difference affect the cell on a long run to receive those signals and act accordingly? Because if it is done in vitro, it will not be adapted to the situation in vivo. And when we put it in vivo, how will the cell respond?
So I don't think there is a general answer to it. Clearly the in vitro response of the cells and the in vitro culture is very different from in vivo. And when we talk about that culturing them in vivo and then putting, say like for example stem cells, putting them back in the patients, again there are a lot of the factors that is involved, and you are correct that many of the times what we get in vitro that does not happen in vivo. The body rejects, the cells don't adapt, in the in vivo conditions, they die, they get cleared off by the immune cells. They don't differentiate the way we want them to differentiate. They form tumors. They give the these graft versus host diseases. So I mean, that is one of the kind of a running challenge in the regenerative medicine, is that how I can prepare the cells in vitro so that when I put them in vivo, they do work the way I want them to do work, right? And as I said, there is no general answer. It is very context specific. In many of the cases, we have lot of success, but not, but there are also many other examples where success is far away.
Oh, okay, thank you.
Thank you, Natik.
I'll read more upon this. Yeah.
Sure.
Hello.
Tanvi, you want to? Okay.
Oh, yeah, sorry.
Yeah, go ahead.
I really enjoyed your talk, sir.
Thank you.
My question to you is that about the mechanical signaling, what are its application in animal tissue culture, and how can it maybe help in enhancing the efficiency of whatever we aim to achieve by doing animal tissue culture? Let's say from a perspective of in purely industrial animal tissue culture.
So like, what do you mean by industrial animal tissue culture? Can you just elaborate a bit?
Okay. So for example, in fermentation, we
fine.
manipulate
Got it.
all the conditions just for our. Okay.
Correct. Yeah. So yes. So again, we have not discussed all those things, but what the cells secrete, right? The secretome. Secretome depends on the substrate stiffness. Substrate stiffness influence the amount of secretome as well as the composition of the secretome, right? So now people are working on both in silico work as well as the experimental work to understand how the cellular metabolism changes when you give them different types of mechanical signals. So now if we want to have a particular kind of, say, industrial production, and if you know that how particular cycle inside the cell or particular metabolism will get changed if you give them, say, shear stress, then you can, work on that. And improve the production rate, production quality, so on and so forth. So, and also why I asked you to elaborate further when you say industrial and the animal cell culture, because there is another aspect is that the cultured meat, right? So that when people are working on this cultured animal meat, again, the scaffold making itself is a both biomaterial aspect in terms of making it edible, making it, enjoyable, but as well as it should have the mechanical properties so that, say, for example, if I want to create the muscle cells should get aligned and should form the particular, give the particular texture.
Okay, I think I got the gist of it. Thank you so much, sir.
Yeah, thank you, Advait, for this nice question.
Good evening, sir.
Hi,
My name is
Sanvi.
Sanvi. Thank you for such an informative session. So my question was that in one of your lectures and seminars that I've seen on YouTube, you've mentioned about preserving the stemness of the stem cells. And in this one experiment where stem cells are cultured on plastic petri plates and then also cultured simultaneously on the polyacrylamide gels. So they are preserved rather better, and the morphology also is, we are able to preserve it. So is that controlling the rigidity itself of the substrate? Is it enough to, preserve the stemness of the cells, or are there any other advancements in the field to be able to for culturing more and more stem cells for a longer term, of research? So,
right.
yeah.
So Sanvi, so yeah, you are talking about our research. I think some slides I already have here in this PPT itself. Yeah, I think she was just for others' interest. She was talking about this research where we have shown that. So when you work with this mesenchymal stem cells for a regenerative application, you need a huge number of cells. However, when you first of all, from the bone marrow, you get only few thousand cells. And when you culture them in plastic in vitro, they go into something known as replicative senescence. They lose their morphology, they lose their differentiation potential, etc. So what we showed that if we cultured them, so this work was done by Sanjay, who has graduated recently, that if you culture them on a soft gel, you can maintain their stemness. So Sanvi, now to answer your question is that maintaining their, so to stop the senescence and to grow in number, that is one aspect that our results showed that the mechanobiology can help. Also to maintain all the differentiation potential, that is another aspect to it. That is also what we have shown. What we need to do and what we have not done yet is that, for example, if I just talk about the mesenchymal stem cells, clinically they are also used for their secretion. So I need to check whether the cells cultured on gel for the long term, whether their secretome is maintained same as the young cells, as well as whether there is any any genetic or epigenetic changes that might have happened, which might be detrimental for the the clinical use. So those check we have not done. We need to do that.
Yeah, thank you so much.
Thank you.
Can we limit the question? I'm really liking the the interaction that's going on here, but in the interest of time, Dr. Majumder is sitting here for 2 hours and he's not also well. So can we limit the questions to like less than 1 minute and make it crisp? Maybe we can club a couple of questions and then
yeah, if there are a common pool of questions, I don't know how to.
Yeah, maybe Parijat, you can read some of the questions that are on the chat board, and then we can go to the people who have raised hands. I
Yeah.
think Ahmad has been
Vinay,
raising.
I can also recommend maybe in the interest of time and given
May
the
I just sorry to interrupt. We can take a few questions, but the another option is that if the students, they can write their question to.
Yes.
Yeah, so that is what you were suggesting, Rajat.
Yes, exactly. And we
can
can
collect all the
those
questions and send it to me. I can give a
Yes.
written reply.
Abhijit,
exactly.
so we can post an Abhijit's email here. You can write to him directly, or you can send questions to us. We can send the questions.
So I am just putting my Gmail ID because my official ID often gets. But you know what I would suggest, that if you can collect the question and send me a full question rather than individual people sending me.
We will do that. We'll try and get all the questions from the students, make sense of it, arrange them, and then send it to you, maybe a Google Doc or something, and we can share that doc with students. So that's easier,
everyone can see everyone's questions.
And your email does not get flooded with so many.
That will be great. Yes.
Okay, so Parijat, do you want to conclude, and we can maybe ask not to give concluding remarks? And I also want to, one second, I also want to call up if any other instructors are present here. There are 150 people. I can't.
Yeah, that's what I was going to say. Let's go. At least we would like to request all the instructors to turn their videos on and say hello to the cohort. And just before we start doing that, I once more would like to thank Abhijit for making time, even when he was not well, sparing 2 hours of his time and talking to all of us and giving such a wonderful lecture. I hope all of you found it inspiring scientifically and otherwise. Yes, that's all. Thanks again.
Thank you, Parijat. As I always say, that we teachers have this thing that when we go to the classroom, we actually get energy. So thank you very much. And I think someone has asked this question: how do we send the question to the team? So maybe you guys can just answer.
Yeah, we will give you guys a little guidance on that. I know, Abhijit, you've been sitting here for long, but last couple of minutes, I would like to invite Nag just to give concluding remarks, and then we can close the call, and then we can have an informal session with the instructors. Students can ask any other questions with the instructors and the course directors. Nag?
Okay, great. Thank you, Vinay. Thank you, Abhijit Majumder. It was great to have you on the platform. And for the first time, I learned about mechanobiology very clearly, the definition, and the way you taught about the emergent property of the cells when they're working together as a tissue, I think was very clear, and it's very, very impressive. And thank you for coming in. Yeah. And yes, so moving on, I would like to thank the course directors here for cell biology and neurobiology. So our veterans are Parijat and Anup for cell biology. They have shaped the course after the haphazard first year syllabus. They have shaped it very well in the previous semester. And this year, Rajan and Sanjeev took it over, and then they want to shape it further. And then they're giving it a different touch this time. And I'm very excited to to see how the course is going to go and the students learning as well. And I would like to thank Aishwarya and Mustafizur, who are course directors for neurobiology as well. And that's very exciting again. And there is a little bit of changes there as well this year. We're learning a lot, the science part, as we move on. And I'm looking forward to that as well. And then I would like to thank all the teaching assistants who are going to be phenomenal and then instrumental in interacting with students and running the courses more smooth. And I look forward to that as well. And I'm very excited to meeting all of them and then talking to them and how we want to do that aspect as well. And a big shout out here is to Garima. And, we always wanted a structured, administrative work on SciencePer, which we've been trying for the past couple of years, but I have a feeling that we've gone much far this year. And, she has been doing a fantastic job and phenomenal with, contacting instructors and students and dealing with all of the work and organizing interviews and Zoom calls and whatnot. Right. Thank you, Garima. It was fantastic. Thank you very much. It's great to have you on SciencePod.
Thank you
And,
so much, ma'am.
and then in the end, I'm coming to the most important part, which are the students. And you are the central focus of SciencePod, and SciencePod is there only for you. Right? Like, we all want to work here for you and then interact with you, teach you as best as we can. Teaching only works well when you ask questions, which you're already doing, right? It has to be two-way street. And we will learn together, as Parijat mentioned before. Please ask a lot of questions, either in the classroom or sending you a question through chat or an email, right? It's always good to have a question, to have it clarified. Not that we all know every answer, but we work on it together, right? So having questions, inquisitive nature to ask questions, why is it happening? How is it happening? When is it happening? Where is it happening? This is all very, very important. That's how you understand a concept. And through this, you can actually learn to approach a problem, right? So the biology is all about, as Rajan mentioned, it's very dynamic system. There is a lot of things going on. We don't understand a lot of it. So asking questions is the best way to go about understanding and also doing research, right? I encourage all of you to ask a ton of questions always and be regular to the classes and then be attentive in, learning. So let's have a fantastic semester this fall, right? Welcome everybody again, and thanks for being part of SciencePod. And in the end, last but not least, the funding agency, which is Lakshmi Mittal Institute, which believed in us for a long time, have been funding us for the past 3 years. And we hope they will continue to do that. But we've been, it's a great thanks to them for, for running this. Yeah. Thank you.
Maybe I can reciprocate a few lines. I would also like to thank Vinay and Nag. It is through their effort that we have this wonderful platform where, we get to interact with all of you. So one round of applause for two of our co-founders.
Thank you, Rajan.
You're humble. Thank you.
So thank you very much. I mean, I was not aware of this particular effort, but it is fantastic to see that, the what you are doing. And I mean, I'm really amazed. And, thank you very much for having me here. It was a great honor.
It's the team again.
It was a pleasure. Thank you.
Yeah,
Thank you, Abhijit.
but it's the teaching staff, right? Like the instructors. It's this. We started with 20 instructors, which was very close network, but now we are 50 almost through friends and bigger network in academic environment. It's been fantastic working with everybody.
Yeah. So
okay.
The recording.
So I may take your leave now then.
Yeah.
Okay. Yeah.
Yeah.
Have a good night. Thank you.
Thank you, Abhijit.
Thank you so much.
Thanks,
Thank you
Abhijit.
so much for your time.
Bye. Take care. Bye.
Thank
please
you.
take
Take
care of your
care. Hope
health.
you feel
Bye.
better soon. Yeah.
Yeah.
Thank you. Bye.
All right. So now the forum is open for at least we can keep it open for maybe 10-15 minutes. We have course directors here, and Nag is also there. Garima is also there. So if you have any questions for us, feel free to ask about the course. But Sanjeev and Rajan, I think we'll also have another session of orientation if the need may be, or
Yes.
just jumping into the direct course.
Yeah. Yeah, I can definitely. We will try and put up a document with the list of instructions, maybe our minimum expectations from students. And we are also trying to this year put up a mentoring team where since we have a lot of students, we don't want them to feel left out. So we'll try and assign, a group of 10 or 15 students to a couple of instructors and TAs so that, they feel involved and, their voices are being heard, because sometimes it will be difficult on a digital platform to give opportunity to everybody to, voice their opinion and talk about what questions or what was interesting. So, we'll try and have a lot of sessions where we can interact with our students. But, I would apologize in advance if we cannot give an opportunity right away, but make sure your questions are noted down somewhere so that we can come back and discuss them later in the interest of time.
Yeah, absolutely.
Yeah,
The
and
course is going to go over. Sorry, Sanjeev. One sec.
no, go ahead, go ahead. You can finish.
Right. The course is going to go for 3 months at least. So you have plenty of opportunity to talk to instructors and teaching assistants and whoever you want to have an interaction with, right? So don't be worried or disappointed if you don't get a chance in one class, because as Vinay said, it's like cohort of 90 students now. So we will have to balance teaching as well as taking questions, right? So we'll have extra sessions where office hours or something, so where your questions will be answered, only question answer sessions, yeah, when the questions are not answered in the classroom. So please write down your questions and take them. Sorry, Sanjeev, go ahead.
Okay, so I just wanted to say before we forget, I think a lot of people still are asking questions related to Abhijit's talk. So maybe Bhargav, can you copy out all the questions for a later, like a document to be made and send it to one of us, like either me, Rajan, Parijat, or Anup. We'll compile it and send it across to Abhijit.
Yes,
And
yes, I
the
will.
other thing, yeah, thanks, Bhargav. Other thing I was about to say is, like Rajan said, we'll have you all the students distributed into groups. So that would happen within the first few sessions. So it would be really great if you can encourage everybody to be there for sure for the first few sessions, so that we can already have you distributed amongst different groups with the TAs. Yeah, that's another request I had before we start.
Yes, and I know people have questions about recorded lectures, and I'm sure Garima will come up with the best way to share this with all of you. So don't worry, we have that covered, and we'll soon get back to you with all of these things. So yes.
Also, the YouTube link will be there, I guess.
Yes, I think so.
Yeah.
Yeah,
Yeah,
so Garima will, if you don't have any others contact, if you can remember GARIMA@sciencepur.org, then you have contact for everyone on the SciencePur platform. So if you need any information, single point of contact is Garima. So you can write an email to her and she will direct your email to the right person. So coming to the recorded lectures, we have a platform called @Folks. We will use that to share the recorded lectures. We also have created spaces, Google Spaces, where you can have like a very real-time exchange of information. All cohort students will be in one space, and Garima will also be there. And then you can ask all the questions if you have any related to your courses. So she will help you there as well. One thing is you need to give your Gmail to Garima to be able to be part of that space. Also, you have to enable that going into your settings. You have to enable the chat settings in your Gmail. Then only you will be able to see chat and the spaces being enabled in your either on your Gmail app on your phone or in the web Gmail application. So that is one important information. And then also, as Rajan and Sanjeev pointed out, we will send out a clear guidance, rather guidelines on and minimum expectations for all of you, so that you will be well informed. And if you have any questions, you can get back to course directors or Garima. And also the chat, Garima can also share the chat history. She will be able to access it. And maybe Garima, you can share that to with Bhargav so that he can send it to the course directors.
Sure, I'll
Yeah,
do
or you
that.
can send it directly to us also. That's fine.
Yeah. So anything else? See, we want this. This is a virtual class environment. This is designed to be virtual class environment. Mitigating the physical borders. We want to see you people. Okay. We don't want to talk to a name that pops up on the screen. We want to see people and talk to them and relate to whom we are talking to and make that personal connection here. So we highly encourage you guys to keep your cameras open and be respectful for the people who are keeping the cameras open and talking to you. And don't forget that all these instructors who are coming here to teach for with a commitment for free. They are not paid on anything, and they love teaching science and they love to communicate with people, and they believe in sharing the knowledge to the next generation to nourish the next generation to be better scientists for the betterment of the society. So please use this opportunity and be respectful to their time as well. And I'm sure you all will benefit from this platform. And we are looking for an exciting semester. And we are always here. If you have any concerns or questions, you know whom to contact, and we'll be at your service. One other important contact for you is you will get that information as well pretty soon, your TAs. So each will be tagged to their TAs. So they're also one immediate contact for you guys pertaining to the course structure. And if you have any questions beyond your TA's purview, you can contact your course directors or Garima. Garima will direct to the right person. All right. If you guys have any questions, we can still answer them. Otherwise, we can maybe Parijat, Sanjeev, Rajan, Anup, if you have any concluding thoughts, remarks, or Garima, go ahead and we'll conclude the call in next 2 minutes.
Okay, so I had some pointers to make. So first of all, welcome everyone. We have an exciting road ahead of us, and I'm pretty sure that we'll come out of it successfully, and you all will achieve your certificate of excellence towards the end. So yeah, this whole journey will be mapped by me for you. And in case there is any trouble, as Vinay said, and your course director said, I am there, I'll be your point of contact. So anytime, any need, just drop me a message on the Google chat or on my email ID, and I'll reply to it as soon as possible. The second point is that we'll have all our classes, like Vinay said, on Zoom. So I want all of you to please update your Zoom. You'll have the schedule of the classes on your Google Calendar. So just make sure that your Google you just have a look at your Google Calendar, and it is with the registered email ID with us, the registered email ID with us will have the schedule of the classes in the Google Calendar. For the neurobiology cohort, the groups have already been divided. Your TAs know, and they will reach out to you within this week, within the coming week. Same for the cell biology. I think the next point is that, like Vinay said, any problems you're facing in academics, your TAs, your course directors, your instructors are the best people to guide you. Anything related to administrative tasks or maybe scheduling of classes or instructors, you can just reach out to me. And one last point is that, like Vinay mentioned, for Google Chat, I would need all of your Gmail IDs. So I see that a lot of people, a lot of students have registered with us with their organizational ID, organizational email ID. So we are not able to accommodate you in the Google space. And this Google Space will be used for coordination and scheduling. Of course, we'll send you important and urgent information through emails. But yes, it will be better if you guys send your email IDs, the Gmail IDs to me, so that I can add all of you in the Google Space. So what I'll do is that you need not send me individual IDs as of now. After this call, or maybe in a couple of days, what I'll do is I'll reach out individually to those whose Gmail ID I do not have. And for all of those students whose Gmail ID I'll add them to a Google Space. So what you need to do is go to your Play Store, or maybe just go to the Google Suite and download the chat app. If you are using your laptop or your computer, you need not download the chat app also. It's just there. There are 9 pointers towards the right hand side in your Gmail. Just click on that. You will have the option of chat. Just go to chat. In the chat screen, you will see spaces option towards the left hand side. So just click on that. You will have a request from me. And if I'm sending you a request, you will receive an email also. So do not worry at all. And all of these instructions will be sent to you later as well. So I will be reaching out to those people who have not registered with us with their Gmail IDs, and then you will definitely be added in the chat. And if in case it is not done by Tuesday end of day IST, please reach out to me personally. I think I am done with what information I wanted to give. So again, welcome everyone, and I'll see you across.
Thank you, Garima. Welcome everyone once again. You have a great weekend, and see you in the regular class sessions.
Yeah.
There is one guy who's raising hand there. What's up?
Sir, can you hear me, sir?
Yeah.
Yes.
Sir, as I can see that there are 122 applicants or students here. Can we meet or can we know the names of the members who are participating in this, or who are attending this meeting? In this case, it will be very fruitful that we can know each other.
So for us to have this introductory session, it will be another almost 3-4 hours. It's like at least 30-40% people have dropped off. So it is actually 200 people cohort for both cell biology and neurobiology. Introducing each one of them will take forever. So what I would suggest is be regular to the classes. You will come to know each one of them and where are they from, because each instructor will ask them, and when they are conversing with them or interacting with them, you will get to know each one of them. So it's better to be more participatory in the class and in your group discussions as well. So that is the best way to know your batchmates.
Yeah, it was a good suggestion. It would have been possible if it was probably last year or the year before, but we have a lot of people here and you will get to know them eventually.
Yeah.
Okay, thank you, sir.
Thank you. Thank you all. You have a wonderful weekend, and excited to have you all once again.
Yeah.
Great. Have a good day. Bye.
See you soon. Bye.
Thanks to you too, sir. Have a nice day.
Thank you, everyone.